final talk in this series of talks on diversity. My name is Tirthankar Bhattacharya, and I happen to be the chair of the Outreach Committee of the Indian Academy of Sciences. This series of talks, the diversity series of talks, was a brainchild of Professor Partha Majumdar, who worked hard to get all the speakers, and it has been a wonderful series. It's organized by the Indian Academy of Sciences, the Academic Trust, in association with the National Council of Science Museums. Today, for the final talk on human history and diversity in India, which is a very interesting topic, we have two speakers, Dr. Madhushree Bandyopadhyay and Dr. Analabha Basu, and it's my pleasant duty to introduce them. Dr. Madhushree Bandyopadhyay started her career as a faculty in the Chemical Engineering Department of Jadavpur University. She found her call, so to say, in prehistory or history and genetics, more precisely population genetics of the Indian subcontinent, perhaps. And she takes great pleasure in unraveling the past, which is clear, otherwise she won't be in this subject. A recent book, Pragitihas, in Bangla, has been widely admired in academic as well as non-academic circles. I personally am looking forward to reading the book. The book also received the Muzaffar Ahmed Memorial Prize in 2021. She also is the editor of a history portal, and again, something that I'm looking forward to. Perhaps I'll soon join it. And so that's Dr. Madhushree Bandyopadhyay. She will speak on the second part of today's talk. So the first speaker today, they will split the time between them. The first speaker today is Dr. Analabha Basu. It's a pleasure to introduce someone I have known for a very long time. Analabha did his MStat in 1998 and PhD in 2004 from ISI, Indian Statistical Institute, under the guidance of Professor Partha Majumdar. He's a young associate professor at the National Institute of Biomedical Genomics. His research interests lie in population genetics, genetic epidemiology, and admixture mapping. He's a fellow of the West Bengal Association of Science and Technology. He received the Young Investigator Award at the 12th International Congress of Human Genetics in 2011. His project with Shayur Karmakar, who is now in US, and Anil Ghosh, again someone I know, found an honorable mention in USPROC 2011. This is a competition for undergraduate projects. So with these two excellent speakers today, no further ado, Analabha, all yours.
Good evening, everybody, and thank you so much, Tirthankar. Thank you for this wonderful introduction. It's a pleasure to meet people whom we know for a long period of time, a bit in these changed times over the internet. So the way in which Madhushree and I planned our talk is that I will mostly deal with the basic premise by which we use genetics in order to decipher about history and prehistory. Without getting into the more technical details of this, I will mostly try to give an intuitive introduction to this, and also give you an introduction to the origin and dispersal of the anatomically modern humans all across the globe. Madhushree then will take it on from there and will lay her emphasis specifically on the Indian subcontinent. So I start with this basic premise that nothing in biology makes sense except seen in the light of evolution, and this is applied for general biology, and this is the title of a paper by Theodor Dobzhansky, and I'm sold to this idea, and this is one of the reasons that it excites me, and this is one of the reasons that I would like to entangle evolution, because it also besides letting us know about our past, besides letting us know about prehistory, it also makes huge sense in terms of biology itself. So just to get very quickly into things, which is just a basic introduction to all of this. This is a picture stolen from the internet about a cartoon of a human cell. So this is the human cell. The human cell is, as you know, is microscopic. You cannot see the human cell in your naked eye. And within this cell lies this nucleus, and within this nucleus are these chromosomes of ours, and these chromosomes are ours is where this fun lies, or the genetic material, or the unit of heredity which lies. So this material is passed on from one generation to another. The parents pass this on to their offsprings, and this is where the information about all our processes, all our being essentially lies. So this is where the genetic material lies. So besides the nucleus, there are these mitochondrions, where there is also a little bit of genetic material. The speciality of a mitochondria is that it is passed on from the mother to the offsprings, and it's a maternal way of inheritance. It's passed on from the mother to the offspring, and only the female offspring would pass this from her generation to the next generation. All right, so so within this nucleus, this chromosomes and the DNA, the way it's packed is a fascinating way in which it is packed into our DNA. So we as humans, we have 23 pairs of chromosome, of which 22 are autosomal, which means that we get one from each parent, and this gives us all kinds of traits that we have. We oftentimes call these as phenotypes, which can be something superficial like our skin color to something which might give us some kinds of diseases, etc. And then there is one pair of chromosome which is called a sex chromosome, where the females have 2 copies of the X chromosome, the males have an X and an Y, where the Y chromosome is passed on from the father to the male offspring. So the members of the same pair of chromosomes are called homologous chromosomes. The amazing thing is the way it's packaged into this. As we have seen that one single cell, you cannot see a single cell by your naked eye. Within this cell is a small nucleus, and within this nucleus is this famous structure, the double helix structured DNA molecule, which is packed into very well packed, very composedly packed into our chromosomes, and it's kept into this chromosomes. And if you take this out, if you take the DNA out and just stretch it and put it in a straight line, then the DNA from one single cell will be somewhere around 6 to 7 feet. So just imagine how fantastic a packaging algorithm is this, where you have packed all of this into this 23 pairs of chromosome, put this into a cell, where the cell is not seen by your naked eye. And not only that, the more fascinating thing is when you need to access any one of these regions of this chromosome, you can very readily access it. So how much of DNA is there in one cell? So this is composed of 4 nucleotides. ATGC is what we say. These are base pairs, and there are approximately about 3 billion base pairs. So just to give you a sense of how big that number is, on the right side of your screen, you can see the number 3 billion, which is essentially 1 million billion. And a base pair is very small. So, but I mean, it's a huge sequence of bases. Essentially, all of us, our genomes, all of these genomes are essentially 3 billion base pairs long. Given that there are so many bases, any 2 individuals randomly chosen in this world is approximately about 99.9% identical. So it's only 1.1% of us which is kind of unique in ourselves and is different from anybody else. Though this as a percentage is a very small number, but because there are so many bases, because there are 1 billion bases, 0.1% of 1 billion is still a very large number. So our uniqueness, in some sense, our uniqueness in terms of the molecules, essentially from this 0.1% of differences that we have with each other. It's also, of course, a combinatorial problem, because it's not necessarily that one of them gives rise to one kinds of traits, but it's a composition of different kinds or different kind of combinatorics by which you can actually get to different kinds of phenotypes. So A, T, G, and C are the short forms of the 4 bases. So these are like the words which make like the letters which make the words in this dictionary of this DNA, and the words have different kinds of meanings. So just to give you a sense of this, because we are almost same to one another, almost our DNAs are identical to one another essentially. So if I take a stretch of my DNA, for instance, so it will be one copy from my father, one copy from my mother. These two are two unrelated individuals in this world, so they will also be same at almost all places except for 0.1%. So there will be only some places where they are slightly different, like here, and rest of the places there can be they are exactly same with one another. So a representation of my genome can be as simple as this, where only the locus and the place where I have something like this is enough which is necessary and sufficient to designate what my genome is, whatever the information content in my genome is. I can remove the rest as long as I have some kind of a reference genome. So the reason that I come from a statistical background and I do all of this is because this genome is our data. And a lot of this is essentially very, very exciting for a statistician or some people who would like data and would like to analyze data. And we'll try and see what we can infer from the different kinds of data that we have. So as I mentioned, this genes gives rise to different kinds of phenotypes. This is one kind of phenotype, which is Kandib. As you see, it gives rise to a huge amount of diversity, and one of the important things that we need to appreciate here is genetic variation gives rise to diversity, which is something which is very, very valuable. Genes are oftentimes very closely associated to diseases, but it can give rise to very strange and interesting kinds of traits. Like if you clasp your hand like this in this picture, then you will immediately see that if you put your right on your left, some of you will feel more comfortable. If you put your left on your right, some of you will feel more comfortable. So which one, whether you feel more comfortable with the left over the right, or whether you feel comfortable with the right over the left is genetically determined to a large extent. There are some people who can roll their tongue like this kid here. Some people cannot roll their tongue. This is again something which is genetically determined to a large extent. All right, so I'll take a pause here, and I'll take you to introduction of this gentleman, because we are going to talk about diversity and evolution. This, of course, is Charles Darwin, and Charles Darwin has been talking about evolution, and he has looked at evolution in a completely revolutionary way in those days. Though he was not the only person who looked at this, there are other people who also helped in this cause of looking this, and this was something which was kind of brewing in the scientific community. So what they looked at is evolution as a process in which he looked at this mostly as bifurcating trees, where there is a common ancestor, and from that common ancestor there are two descendants, and the descendants generally are species, and the species, they diverge from their common ancestors and go different ways and give rise to different kinds of species. So it's this kind of representation still remains very, very valid in terms of understanding of evolution, and he has looked at mostly morphology and phenotype and songs and other fossil records, and tried to put all of this into a tree of life, which essentially is trying to build this tree where species are connected to one another through common ancestors, eventually giving rise to the tree of life. So how do we connect genetics with respect to this? So here is our basic premise. So here is present time, this is past, and time is like going in this direction. And in the past, we have a common ancestor from which there are two descendants. And why is genetics important here? Genetics is important because If you think of this time, then these two species are very, very similar in terms of their genetic makeup, say. And then at some point of time, there are random changes as they pass on the genes from one generation to another. There are random changes or mutations, as we say this. Mutations happen over time, and because the genome is large, mutations separate out these species. So there are these species which are more distant to one another are separated for many mutations, and species which are closer to one another are separated with fewer number of mutations. So these random changes are mutation events, which are observable changes in the genome. They give rise to differences in species, and just by counting the number of mutations, if there is a regularity in which these mutations occur, if there is a basic regularity or basic like a clock-like phenomenon back in the background in which these mutations occur, then we can use this number of mutation or number of observable changes between the species to try and figure out how different or how distant from the common ancestor to the two species of these types are. So this is something which was put in as a hypothesis, and E. Margolish in 1963, she looked at the cytochrome C, which is essentially a protein, not necessarily DNA in those times, and looked at proteins in different species and found that they kind of fitted well with the ways in which evolution was designed or evolution was predicted. So just to put things in perspective, this is me. This is a picture of myself, and I'm pretty close to a chimpanzee in terms of our distance, in terms of our species distance. And whoever tells you what, we are pretty distant from cows. So what will happen is that if we have segments of our genome, then my genome and the chimpanzee genome will be generally pretty close or very similar, except for, say, one or two places, whereas the difference with the cow will be much distant. So if you put that in perspective, in a larger scale, this is how it was calibrated. This is an old paper where people have put this evidence with respect to archaeological records. So they have dated archaeological fossil remains, and they have calculated molecular divergence time using sequences, and they see that there's a very strong correlation between them. So as we go along and technologies develop and we have more resolution, what we can do is we can essentially differentiate between these two individuals whom, if you recognize, these are two brothers and sisters in one of those films, and they are brothers and sisters, and that's the reason they are much more closely related to one another. So they will have fewer number of differences in the genome compared to a Masai Mara who lives in Africa will have many more differences. Using this, what we have done, or geneticists have done, is at some point of time they have looked into the evolution of anatomically modern humans, and they looked into anatomically modern humans from all over the world. What they found is that they essentially have constructed a tree. And in the tree, they have counted distances. And what they found is if you take populations from all across the world, so these are populations from all across the world, they have a common ancestor somewhere here, okay? And everybody here who ends with an open circle here, these are people who are from Africa. So all Africans, they go very, very deep and close to their common ancestor of this entire tree. And all non-Africans have a common ancestry somewhere here, which is much more recent in terms of timing. So what they infer from here, this is using some kind of mitochondrial sequence data, is that the anatomically modern humans started their journey all in Africa. Not only they started their journey all in Africa, if we have been on this earth as anatomically modern humans for, say, for a football match, then the first half has been played entirely in Africa. So half of the times we are on this world, the first half has been entirely in Africa. And after that, there has been there might have been other successful, but there is one successful out of Africa migration by a very small number of individuals who have been successful in setting up colonies across the world. They moved out of Africa and set up colonies all across the world, and they conquered the world in a very, very quick time point. So this is what they use, a very small segment of which is called a hypervariable segment of mitochondria. This is 1991, and in 2000 they use the complete mitochondria to come up with very similar kind of inferences, where on anything on the top of this vertical of this horizontal line are non-Africans. So if you look at the genomes of non-Africans, they have a common ancestor somewhere here, marked by this star. But on this side are all Africans, and you see that the African genomes go very, very deep and close to their ancestors, close to their most recent common ancestors of all humanity. So this individual is called as a mitochondrial Eve, and this is also like hanging by our outgroup of our closest known ancestor or closest known cousin, which is a sheep, who was the closest known cousin at that point of time. So the great human exodus, the story goes like this, that it all started somewhere in sub-Saharan Africa, or for some people they say that it's somewhere further south. But the main story is there have been movements all across Africa, but modern humans in around 200,000 years, depending on who you talk to, they originated somewhere in sub-Saharan Africa, then moved around in Africa for a very long period of time. In around 60,000 years from before present, they moved out, and they moved out to different parts of the world. They moved out in different directions. One of the first places to be where people started living, there has been very strong archaeological evidence that in fact by 50,000 years there has been people who lived in Australia. And if you move to Australia, obviously, however you move, you have to move to India as well. So India is one of these places which has been inhabited very, very long time back. So when we moved out, we do not move into a vacuum. There were archaic humans, hominids, our hominid cousins like Neanderthals, etc., who were living there. And there has been many kinds of coevolution support of the out of Africa hypothesis. So this is human mitochondrial DNA. If you look at this tree, see how similar this tree looks with respect to the tuberculosis, Mycobacterium tuberculosis tree, and this is essentially because tuberculosis is a very old bacteria which we have been carrying along from time immemorial, and the African tuberculosis bacteria has more variation, exactly like the African humans have more variation. So this is like the graph of the time travel of anatomically modern humans. We started somewhere in sub-Saharan Africa, and we moved out to different directions. And when we moved out, we lived Australia very, very early. Australia was inhabited before Europe was inhabited for the time of Europe was inhabited, although the geographical distance here is small. One thing which we need to keep in consideration is when people were moving out, it was ice age, so there was a lot of water which was trapped all here, which means that the seashores were shallower, which means people could actually walk all the way up to the tip of Indonesia to get to Australia, but still you need some kind of sea craft to get there. So this is how the if you think of this is how the Indian subcontinent also looks like. So as you see that India and Sri Lanka, they were very, very closely connected, and not only that, Andaman was also connected. So you could actually walk along the coastlines all the way along the Andamans to somewhere which goes into South and Southeast Asia. So the great thing in this is that India lies, of course, in the center stage of evolution. And one thing which you can think of right now is genetics, which deals with extant individuals or individuals who are living today, and trying to infer from their data has an age in inference about history. Because if you think about the peopling of whether there has been a peopling through this direction, these places have now are under sea. So archaeologists do not really have a chance to dig it out from here and kind of come up with an inference with respect to this. But if you use genetic data, because it's written in our genes and people are still living, you can still reconstruct history of whether there has been movements along this coastlines into Australia or not. So this is how the lines are made. So there are two hypothesized way of out of Africa migration. There's one out of Africa migration through the north. Some people say that it has been out of Africa migration through the southern exit route as well. So these are timelines. These timelines of process estimates with some kind of some kind of a standard deviation, but basically the chronology of the timeline fits well. That we have been in Africa for a very long period of time, moved out of Africa, stayed here for a long period of time, traveled to this direction towards Australia very early. Europe was populated a little later than the Americas. The North and the South America was the last two continents to be populated, and South America was populated only about 30,000 years ago. So let's change gear a little bit here, and as we have explained, as we have discussed, that genetics is able to decipher from extant genomes. So it looks at the genomes of present-day individuals and try and reconstruct history by looking at differences. And there are also other methods, other sophisticated methods by which it does this. But is there a possibility that you can actually look into old fossil remains or old remains of skeletal remains and try and infer about history, like archaeologists do? Archaeologists dig into the past. Really, they dig into the past. So can genetics do any better? So there are two individuals. This is Alan Wilson, who did all of this. This out of Africa came out of Alan Wilson's lab, and this is Svante Pääbo, two of the protagonists who thought that the DNA is essentially a very stable molecule. So although these individuals who have died, or individuals for whom you have skeleton remains today, though these individuals are no longer living, but their DNA might still be intact, because that's a molecule. You can still get the DNA and get the information out of the DNA. So in 1984, as early as 1984, this is Alan Wilson, who tried to do a DNA sequence of quagga, which is an extinct member of the horse family. And Svante tried to do this with ancient Egyptian mummy. So they both tried their hands with this, and they thought they were successful in this, though both of them acknowledged that they were totally wrong in doing that, wrong in inferring about this. And after that, there was a huge excitement, and everybody thought that they have got ancient DNA out of different kinds of remains, starting from quagga to mummy, to some people said that they got DNA out of dinosaurs. So if you think about expectation, the expectation was rocketed and then eventually it tapered down. Eventually people's delusionment got shed because people got a reality check. And in what happened later in 1997 is Svante could actually get DNA out of Neanderthal sequences. So Neanderthals were our close remains cousins who lived about 40,000 years ago in places in Europe, and he could get these bones, and from these bones he could extract material from which he could extract DNA, and he could actually get the DNAs and sequenced. So in 1997, you could get mitochondrial DNA out of it, and this was the breakthrough of the year 1997. What we could show is, if you compare any two humans, they have a fewer number of differences in their mitochondria. If you compare a human with a Neanderthal, they have much larger number of differences. If you compare a human and chimp, they of course had a much larger number of differences. So if you do some kind of a unitary method, if this is like a 5 million years of separation between a human and a chimp, then the human and the Neanderthal difference will be somewhere around 1 million. And he established that Neanderthals and humans are probably different kinds of species, and they are separated about 1 million years ago. So there have been changes in the sequencing technology, and what has happened in the recent years, in 2009, 2010, in all along this time, is these molecules, which were required in large quantities for doing some kind of genetic analysis, now you can use very little amount of these molecules to sequence this entire thing. So now what we have is we have a draft sequence of the entire Neanderthal genome. So we can actually do this for an entire Neanderthal genome. And the fascinating thing about this is, when they were looking at many more Neanderthal genomes, they came up with a genome of an individual who's not like a Neanderthal. The skeleton looked like a Neanderthal. They share a cave which is inhabited by Neanderthals, but within the cave they found a genome which is very different, of a different species, which is a closer cousin of a Neanderthal, and it was found in a Denisova cave, and they call that species as Denisova, which is closely related to the Neanderthal and is a close cousin of our modern humans. So this was our understanding of evolution, where a common ancestor from which orangutan gave rise to a different species, and then gorillas, and then chimpanzees, and then we were humans. Now what we have is Denisovans and Neanderthals, which are much closer, and these are extant individuals for whom we can still have our genomes and reconstruct our genomic trees. And now we have this, for which, though these people are no longer here, the Denisovans or the Neanderthals, but we still have their DNA sequence to reconstruct and investigate about their genomic, investigate with the genomic data. What it gives us is that the reconstruction of human DNA, human history using DNA, is not limited to extant individuals only. And DNA from individuals who lived and died long back are now available, and those can also be incorporated. And that's one of the things which people have been doing very, very extensively all throughout. They're trying to get DNA out of all kinds of molecules from all across the world. And one of the greatest recent breakthroughs in that has been that in hot and humid climates along this place, you understand that this is the northwestern part of India, and this is the Indus Valley. This is Rakhigarhi, and there was a skeleton in Rakhigarhi from which people could actually get DNA molecule out, and they could sequence, and they can infer a lot about the history directly from people who used to live in who probably used to live in the Indus Valley. So I would end with two notes on evolution before I go. One, of course, is that somebody from a seat of power has once said that evolution is wrong because nobody has seen a chimpanzee to change into a humans. This is fueled by cartoons like this or this, or even sometimes people write things like this, but a better nuanced understanding of evolution is more like this, which is the Darwinian way of understanding evolution, which is that there are common ancestors from which there are different species which generally bifurcate, and evolution is generally a continuous trial and error kind of a method, in which here we have our closest living cousins, the chimpanzees, and here are the Homo sapiens or the anatomically modern humans. But there has been so many trials in between of different hominids who lived for some period of time. There are some of these old names which we know, say Australopithecus, Homo erectus. They have lived for a long period of time. We have their fossil remains, we have their artifacts, but we do not see these people around. So evolution works in this way, and this is a much more nuanced way of looking at evolution. It is not like a chimpanzee walking into becoming a human. It's more like a bifurcating tree where it separates. It's like a species. Some species live for long, some adapt better with the environment and live for long, some do not. The other thing which I would like to emphasize here is Homo sapiens sapiens. We are the proud Homo sapiens sapiens. We are the latest people who are here. We are essentially the least adapted to this world, and we have been the cause of extinction of many species. But we should keep in mind that we have been in this world for a very little period of time, and we haven't adapted to it the way in which there are other species which have adapted. Actually, we have adapted the least to this environment, to this world, compared to anybody else. So if we continue doing this, what we are going to do is, it really doesn't matter the nature or the world, but we are going to make ourselves extinct very soon. So with that, I'll stop here, and I think Professor will probably talk more about, emphasizing more about the diversity and history in the Indian subcontinent.